In my previous video, I discussed about the composition of nucleotides, what is DNA, compositions of DNA, and we have already learned about what is nitrogenous base, what is pentose sugar, and what is phosphate group, and why Watson and Crick model is most accepted model and why they get Nobel Prize for their discovery. Now students, I am going to explain the main features of Watson and Crick model of DNA. Student, first of all I want to tell one thing. Student, I summarize the total model in 10 vital points. Student, please concentrate now because these 10 points will very important for a biological science student, because as a life science or biological science student you must know about the structure of DNA in detail, as it is very important for your CHSE and medical examinations. Student, it is very important for you for your upcoming board exam and in any all India medical examinations. So students, now I am starting. Now point number 1. The point number 1 is DNA molecule consists of two polynucleotide chains or strands. That means it consists of two strands. Here is one strand and it is another strand. If you take it in 2D structures, it will look like this. It is one strand, it is one strand. It is one strand and it is another strand, like this. If you see that in a 3D model, it will look like this. This is one strand or one polynucleotide chain, and this is another polynucleotide chain. So, the first point is DNA molecule consists of two polynucleotide chains or strand. The two strands are like this. It is one strand and it is another strand. It is one strand and it is another strand, like this. This is one strand, this is one strand. Okay guys, next the number 2 point is the two strands or the strands, these two strands are spirally twisted around each other. These are spirally twisted. These two strands or these two polynucleotide chains are spirally twisted around each other, around a common axis, like this. If it is a common axis, it is twisted around each other on a common axis, like this. First point, DNA molecule consists of two polynucleotide chains and spirally twisted around each other on a common axis, like this. Like a spiral binding, it is spirally twisted. The two strands are spirally twisted around each other on a or around a common axis. Next, number 3. The two strands are anti-parallel, means one if goes in this direction, another goes in that direction. One is running in this direction and another runs opposite direction. You know, as a science students, you know about what is the meaning of anti-parallel. Anti-parallel means one if goes in this direction, other goes in this direction, like this. And if one stand's starting point is 3 dash, its ending point is 5 dash. Like that, if it is anti-parallel, it is 3 dash and it is 5 dash. So, 3 dash end of one chain, 3 dash end of one chain facing 5 dash end of the other, 3 dash end of one chain facing 5 dash end of other strands. Number 4. Number 4 tells that the two strands are held together by hydrogen bonds. Means these two strands are held together by hydrogen. Due to hydrogen bonds, they are bonded with each others. Between the purine and pyrimidines, means you know purines, pyrimidines. In my previous video also I will tell about what is purine and pyrimidines. Purines are adenine guanines and pyrimidines are cytosine, thiamine and uracil, and uracil is only present in case of RNA, but it is not in case of DNA. So in our DNA we will just know about purine and pyrimidines, means adenine and guanine, thiamine and cytosine. We'll never discuss about uracil in case of DNA because uracil is only present in case of RNA. So, the fourth point tells that the two strands are held together by hydrogen bonds. If you take that in a 2D model, it will look like this, guys. In a 2D model, these two strands are held together by hydrogen bonds. If you take that in a 3D model, it will look like this. Hydrogen bonds are present in between purines and pyrimidines, which bind the two strands closer to each other. Once again, the two strands are held together by hydrogen bonds between the purine and pyrimidines, base of the opposite strands. If one is purine, if this side purine is present, in other side pyrimidine. These purines and pyrimidines are held together by hydrogen bonds. The two strands are held together by hydrogen bonds between purines and pyrimidines, base of the opposite strand. If these strands contain purine, that opposite strand contain pyrimidines, and purine and pyrimidines, they are between purines and pyrimidines, there a hydrogen bond is present. If you see that in 2D, it will look like this. If you see in 3D, it will look like this. There is hydrogen bonds between adjacent purine and pyrimidine bases of two strands. Now number 5. A always pair with T, means adenine always pair with thiamine by two hydrogen bonds like this. If you see that in a 2D, in between adenine and thiamine, two hydrogen bonds are present. Two hydrogen bonds between adenine and thiamine. Then guanine always pair with cytosine by three hydrogen bonds like this. Cytosine binds with guanine with three hydrogen bonds like this, guys. They are present three hydrogen bonds like this. 1, 2, 3. Three hydrogen bonds between cytosine and guanine, and two hydrogen bonds between adenine and thiamine. So number 5 points tells A always pair with T by two hydrogen bonds, and G always pair with C by three hydrogen bonds. Like this, guys. If you see that in a 2D, in between A and T, two hydrogen bonds. Between C and G, three hydrogen bonds. If you see that in a 3D model, it will look like this. In between A and T, means like this. In between A and T, two hydrogen bonds are present, and it between C and G, like this, three hydrogen bonds. If you see that, it will look like this, guys. 1, 2, 3. Between cytosine and guanine, three hydrogen bonds. Between adenine and thiamine, two hydrogen bonds. It is called complementary base pairing, means the The bonds which binds adenine and thymine, cytosine and guanine of two strands. It is called complementary base pairing rule because the two strands are complementary to one another. Due to hydrogen bonds which are present in between the two strands, and the two strands are complementary to one another, and in between A and T two hydrogen bonds are present, and in between C and G three hydrogen and bonds are present. This is called complementary base pairing rule because the two strands are complementary to one another. Next, number 6. The diameter of DNA is 20 nm or 20 angstrom. The diameter means the distance between the two strands. The distance between the two strands is 20 angstrom. Like this. If you see that in a 2D, this is the distance between the two strands or diameter. If you see that in a 3D, it will look as this is the distance between. This is called distance is 20 angstrom, or you can call it as diameter also, or you can write it as distance between two strands is 20 angstrom.